Thank you. I'm very pleased to be here, and I apologize that I can't talk in Swedish. I'm sorry. So I'm a visual neurophysiologist, which means I try to figure out how we see by recording from neurons at various stages in the visual system. But I've come to realize that artists also try to figure out how we see. And many works of art are experiments in vision, and many works of art are famous because they reveal important things about how we see. Now, I know that there are a lot of works of art that reveal important things about human culture or human psychology, and even though those are important, I'm not going to talk about them because I work on low-level vision. I'm just going to talk about works of art that reveal things about how we see. And the first thing I want to talk about is the fact that artists use lines to represent scenes and things, and we're perfectly content with that. We see the objects that the artist conveys with a line drawing, even though there are no lines in a real scene. In a real scene, there are contours and discontinuities, not lines, and yet artists invariably use lines to represent contours. And the reason this works so well that I have to tell you that it's not what you see is because of the way our brain processes visual information. Our visuals, Steve Kufler figured this out. Steve Kufler was Torsten and David's mentor. He figured out that if you shine a spot of light on the retina, you get a barrage of action potentials, neuronal signals. But what was peculiar was that when he shined a bigger spot of light on the retina, he got a worse response. And Steve figured out that that's because retinal ganglion cells are excited by light in one part of the visual field, but they're inhibited by light in the immediate surround. So when he shined an annulus of light on the retina, he got a suppression of firing. We call this center surround antagonism. It's the most fundamental stage in visual information processing. And in case you don't believe me that your visual system is full of center surround cells, I hope you see all the little gray dots everywhere where you're not looking. And don't worry about why it's where you're not looking, that's postgraduate. You see the little gray dots, right? Okay, you see the gray dots because center surround cells at the intersections have more white in their inhibitory surrounds than center surround cells at the cross pieces. So these neurons are less active, and therefore they look darker than neurons along the cross pieces. There's an extra synapse involved in calculating this surround inhibition, an extra step, so there's a tiny delay between the center and the surround. You need to keep up with all these people who are sneaking in late with the glasses, okay? So do you see the scintillating little delay in your center surround antagonism? So if you take anything away from this talk, I hope you will take away the message that vision is information processing. It doesn't do you any good to send a high-resolution image to the back of your brain because there is nobody up there to look at an image. All you have is neurons. All they can do is fire or not fire. Therefore, these computations must be simple. All they can do is fire or not fire. Local, they're neurons, they're little cells. And they're often opponent because neurons can either excite each other or inhibit each other. And they often inhibit each other. So if you have an image like this, white on one side, dark on the other, center surround cells over here are inhibited, center surround cells over here are not activated. It's only center surround cells at the border, the discontinuity, where there's an imbalance between the center and the surround where you get a signal. So center surround cells turn a contour into a line drawing. And this is efficient. This is information processing. Because if you've ever tried to save an image like this, white on one side, dark on the other, on your computer, if you save it as a TIFF file, it's a really big file because a TIFF file saves the value of every pixel in the image. But if you use an image compression program and save it as a JPEG file, it's a much smaller file, right? That's because JPEG does exactly what your visual system does. It codes where the discontinuities are, what the sign of the contrast is across the discontinuity, and it doesn't bother sending information about parts of the image where nothing is happening, where there's no information. And this is just to remind me about how efficient information processing can be in a line drawing, but it also allows me to point out what color are these lines? This is not a hard question. Black. Oh, thank you. What color is this screen? White. So you know that the amount of light coming to your eye from that black line is the same as the amount, because that projector can't project blackness, right? All it can project is light. So the same amount of light is coming to your eye from the black line as the white screen. Your visual system does not care how much light there is at any point in the visual scene. What it cares about is contrast, because its first step is center surround, which basically tells you what the contrast is. Your visual system maps the visual world onto visual cortex like a map. It's systematic. So nearby cells in visual cortex are coding information about nearby points in visual space. So center surround's antagonism is local in visual space, which gives you all kinds of contrast. Oh, I forgot to say this. Long-range connections in cortex are always excitatory. Inhibitory neurons are always local. So contrast, center surround antagonism, is local in visual space. And I told you about surround antagonism in luminance, which is why you see these little gray dots as different, but they're not. These are all identical gray dots. It's the background that I've changed in luminance, and you perceive a difference in the gray dots because of surround antagonism. So luminance is locally, spatially locally antagonistic or opponent. Contrast is locally opponent. These sets of checks in the center are identical, but these look higher contrast than these because these are surrounded by high contrast. These are surrounded by low contrast. Orientation is locally opponent in cortex, so the center lines are vertical, but they look off vertical because of opponency and orientation. Motion is locally opponent. That red dot is moving vertically. I hope it doesn't look like it's moving vertically, but I guarantee you it is because of the surround motion. Color is locally opponent. So all these little gray rings are identical, but these look more yellow, that is less blue, and these look more blue, less yellow, because of the change in the background. And I know they're identical because I made this. Here's a little illusion based on surround opponency and color. If you look at that little black dot for just a second, keep in mind that what you're looking at is white letters, and I hope you'll get a yellow after image from white letters because of the surround opponency and color. And of course artists know this. Monet painted a blue shadow with yellow highlights. So vision is information processing, and these computations are simple and local and opponent, even for something as complicated as depth perception. Now, you would think that depth perception would be something that would involve the whole visual field and wouldn't be local or opponent, but it actually is. So you look out on the world and it looks vividly three-dimensional, right? Do you see the world as three-dimensional? Okay, I do too, and I know the world is three-dimensional, but you see it as three-dimensional starting with 2 flat retinal images. That's all you get. 2 flat retinal images. And from 2 flat retinal images, you compute this impression of distance and depth. You don't have sonar or anything. You have to calculate distance and depth. And you do this by using a number of cues. You use relative motion, so things that are near move more than things that are far away. I think this has to be urged to do this. You get a sense of depth from relative motion. You get depth from shading, perspective, occlusion, what's in front of something else, and stereopsis, which I'll talk about at the end. So you can, if you're going to make an image, many of you would like to make an image using computer graphics, you can pay a lot of money for what's called a ray tracing program, which will guarantee you that the light that shines on some image in your computer graphics program will reflect light in a physically plausible way. And you don't need to do this because nobody goes around making sure the laws of physics are being obeyed. It's kind of a given that they're going to be obeyed. And artists have for centuries ignored the laws of physics because our visual systems don't care, and we compute distance and depth starting quite locally in visual space. So you don't have to have the whole image be internally consistent. So when painters started making shadows, they often had shadows going in orthogonal directions. Here's a nice vase of flowers casting a shadow towards the sun. This is a painting at the Fogg Museum at Harvard of the Piazza San Marco. and the shadows on this side of the painting go this way, and the shadows on this side of the painting go that way. So I was doing a little interview at the Fogg Museum one day, and I pointed this out, and the curator came over and said, well, that's because the light bounces off this building and casts shadow. So I went to the Piazza San Marco, and there is no time of day at which the shadows go in two different directions. Nobody cares. You would think that this thing would cast a shadow, but it doesn't. Here's a manger where the roof is seen from above over here and from below over here. And you wouldn't expect halos to obey the laws of physics necessarily, but you'd at least expect them to be consistent in whether they occlude the person whose head they're around. Magritte plays with the laws of occlusion. You find a painting with a mirror in it, I guarantee you it's not obeying the laws of physics. If this lady were looking at herself, which is what the painting would have you think she's doing, you, the viewer, wouldn't see her face, right? That's just not the way it works. Any mirror painting with a mirror doesn't make any sense. Even Vermeer. You think this brass bowl looks nice and shiny, and you think it looks shiny because it's reflecting the tablecloth, but if you look closely, you'll see that what it's reflecting has nothing to do with what it's on top of. Something else that artists figured out long before us neuroscientists is that color and luminance, and don't worry, I'm going to define luminance, color and luminance do different things for you as a painter and as a viewer. So we have three types of cones in our retinas: short, middle, and long, or blue, green, and red. We see color by opponency, by subtracting the activity in the different cone types. We see luminance by simply summing the activity in our different cone types. So in this image, this is the color version, this is the luminance version. And most color contrast borders also carry a luminance contrast, but you can have color contrast borders that have no luminance contrast. And when you do, peculiar things happen. And I think many artists have figured this out. This is Monet's Impression Sunrise. It's at the Marmottan in Paris. And the sun in this painting, not in this version of it, unfortunately, but in the original, is really shimmery and bright. And it looks hot and cold and light and dark at the same time, but it looks just brilliant. And you would think, of course, that the sun should be brighter, should have more luminance than the gray sky background, but I once got a small grant from Harvard to go to Paris and measure the luminance in Impression Sunrise, and it turns out that even though it looks bright, it's exactly the same luminance as the gray background. And I think it's because it's the same luminance that it has this eerie, shimmery quality. And that's because your visual system, and the primate visual system in general, is divided into these, after it gets to primary visual cortex, is divided into these two major processing streams, a dorsal and a ventral processing stream. And we know that these two processing streams do different things because of what happens to people who get lesions in parts of their visual system. So people who get lesions in the ventral part of their visual system get what are called agnosias. They can't recognize things. So they can see them, they can grab them, they can copy them, but they just can't say what they are anymore. So we call this the what system because it seems to be for recognizing what objects are. This part of our visual system is evolutionarily new. It's a primate add-on to the basic mammalian visual system. And our dorsal stream is more like what other mammals have for their entire visual system. And it carries information about motion, depth, spatial organization, and figure ground segregation. It allows an animal to navigate through its environment and to see things that are moving, whether they're predators or prey, and just get around. But other mammals are just not as good as we are at recognizing objects visually. So here are some drawings made by patients with parietal lobe lesions, with dorsal stream lesions. Some people call it the where system because it carries information about spatial organization. Some people call it the how system because it's sort of how you interact with your environment. In any case, these patients were asked to draw a bicycle, a bicycle, a cross, and a clock. So you can see that these patients got all the bits right, but the bits are in the wrong spatial organization, kind of like a cubist drawing. Not to say that Picasso had a parietal lobe lesion, but rather that Picasso figured out how distinct where something is and what it is can be. But what's important for what I've been talking about now is the fact that our where system, our dorsal stream, is, like most mammals, colorblind. So there's a whole part of your visual system that sees the world in shades of gray. So if you had a red spot on a green background and you took a black and white photograph of it, you get a gray spot on a gray background. This is the way your where system sees the world. And you can imagine that you could change the brightness of the red spot in such a way that the two grays would be identical. At that point, your where system can't see it, even though it's highly visible to your what system. And for me, this one has that kind of shimmery quality, and maybe this one. This weird quality of seeming to spread into the background and be light and dark at the same time. So I want to try to convince you that your ability to see depth, spatial organization, and figure ground segregation are carried by a colorblind part of your visual system. And to do this, I need to turn the lights down. Lights off. Okay. Our projectionist is going to have a conniption fit, but that just, it really won't, I won't do this. So here the red spots are brighter than the green, and this thing should look three-dimensional. I hope it looks three-dimensional. Obviously it's not. And whether the red is brighter than the green or darker than the green, it looks three-dimensional. But as I go through equal luminance, I hate it when it does that. Come on. As I have to set it up again. Nope, that's the wrong one. Sorry. I forget which way I have to go. Okay. Now, when the red is brighter than the green, it's three-dimensional. When it's darker, and now as I go through equal luminance, I hope it gets flat and squishy, and then three-dimensional. Oh, maybe that's a little flat and squishy. Stop it. So you have to work fast with this program. All right. There, three-dimensional, flat and squishy, three-dimensional. How fast can I do this? Flat and squishy, and three-dimensional. There, I did it. All right. Whew. And now I can we leave these off for just a few more seconds? I know the projectionist doesn't like this. So I hope you see that the artist has used a lot of cues about depth in terms of perspective and shading, but I hope you can make this look more three-dimensional by closing one eye and looking through the red lens of your glasses. And I hope this will now look much more three-dimensional. Close one eye, look through the red lens. Now put the glasses down. So I hope it looks more three-dimensional when you introduce a luminance contrast by blocking the blue light. Here's another version. Don't use the glasses yet. Just look at it and think about how much depth do I see from perspective, how much depth from shading. Can I even figure out what this image is doing? Can I do figure ground segregation? And then close one eye, look through the red lens, and see if this doesn't look more three-dimensional and make more sense. There. Monet painted a whole bunch of versions of this cathedral in Rouen, and they're all about how the light bounces off this beige stone building with the depth. All the information about the depth in the door and the rose window are there in the luminance version. But in some of them, he was studying how light bounces off things. This is one we have at the MFA in Boston, and it looks flat and shimmery. And I think it looks flat and shimmery because Monet used colors that were just about equal luminant, which you can see in the luminance version over there on the right. And here's another one that I found. This is, don't use your glasses yet, this is Lichtenstein's print of the Rouen Cathedral, and it doesn't look like anything, right? Until you close one eye and look through the red lens, and there is the Rouen Cathedral. Andy Warhol played with this. He has a poster of the Brooklyn Bridge. Half of it is in two colors that are approximately equal luminant, and it doesn't look much like the Brooklyn Bridge, and it doesn't look particularly three-dimensional. But here, The top version has a luminance contrast, and it looks much more three-dimensional and makes much more sense than the bottom version, even though it's exactly the same image. And for those of you of my generation, this is Gracie Slick. Doesn't look like Gracie Slick in the equiluminant version, but in the luminance contrast version, it does. So if your ability to see depth from shading is carried by a colorblind part of your visual system, the corollary is that it doesn't matter what color you use as long as the luminance is appropriate. And of course artists figured this out. This is André Derain's portrait of Matisse. If you knew Matisse, you'd recognize him instantly, even though his face is made up of planes of completely bizarre colors. There's no way you could get colors like that. And that's because even though these colors are bizarre, their luminance is exactly right to convey the shape of Matisse's face. And Matisse's Lady in a Hat, when he first produced it, everybody was all upset about it because he was doing what Picasso said. He was using colors symbolically, but the reality, the shape of her face, is there in the luminance alone. So I hope I've convinced you that your ability to see depth and spatial organization is colorblind. Now I want to try to convince you that your ability to see motion is colorblind. And this one I don't need the lights out for, thank goodness. Let's see, I need to slow it down. There. Now, if the green, so these two sets of stripes are always moving at the same speed, but if the green is brighter than the red or darker than the red, they seem to be moving at the same speed. But if you look off to this side and think about the speed, and I can get the stripes on the left to be approximately equiluminant, I hope you get the feeling, don't look at the center, I hope you get the feeling that the stripes on the left are moving slower than the stripes on the right, because they are a suboptimal stimulus for your motion system, because they're approximately, I hope, equiluminant. If your ability to see motion is colorblind, then your ability to see that something is stationary is colorblind. And art historians carry on about Monet's ability to achieve a sense of motion in many of his paintings. I think he did this, accomplished this, by using pairs of colors that were approximately equiluminant. So the poppies are just about the same luminance as the background, and so their position is indeterminate. They can seem to sway in the breeze. Monet never said explicitly that he was doing this, even though he achieved it a lot of times. But I know that Mondrian thought he was doing this because the title of this painting is Broadway Boogie Woogie. The original's at the MoMA in New York. It's about this big across, and it's absolutely nauseating how much it jumps around. And I think it jumps around because your what system can see the yellow checks, but your where system can't, so their position is indeterminate and they can seem to move. Advertisers do this all the time. They use equiluminant text to catch your attention because it has this jittery quality, and it also is hard to read, which I'm going to talk about at the end. So I hope I've convinced you that your ability to see motion, depth, and spatial organization are colorblind and calculated locally in visual cortex. Now I want to try to convince you that your color perception is low resolution. Of course, artists figured this out first. Artists who do watercolors or pastels will often have an outline with a color that, you know, they don't color inside the lines. You don't have to color inside the lines because the color simply adheres to whatever contour it can find, or your visual system can find to assign it to. It's a technique that's very commonly used in art. Here's an illusion based on this observation. I hope you see the donut as yellower than the whole or the background. Do you? It's not. I made this. I mean, I see it as yellow too, but it's not. This is just a yellow line inside a purple line. Yet this donut looks much yellower than the whole or the background. Because your color resolution is low, your visual system lets the color spread until it hits a border. And now I'm going to use some after image to generate some blurry colors so that you can watch the color spread till it hits a border. What I want you to do is look at that intersection, okay? And then I'm going to show you an achromatic field, okay? So now look at the intersection again, and this time do you see a stronger color after image in this achromatic image? And some of you are saying, well, yeah. Some of you are saying, I thought this was a color image. Others of you are saying, well, of course it was better because you did it twice. No, that's not why. Look at the intersection, evaluate the after image. Look at the intersection, evaluate the after image. It's stronger when there's an outline to constrain the colors. Now look at the intersection and see if you can get the color to fill a different shape. And look at the intersection and watch the color just squirt out the hole. So I wrote this book about art, and while I was writing this book, my editor said that it was obvious I knew a lot about vision, and I do, but it was equally obvious I knew nothing about art history. And so he told me I had to read a book on art history. So I got Gombrich, which is a very good book, and I got to the Renaissance, and I got stuck. Because Gombrich pointed out that there is a reason why everybody loves this painting. It's uncanny how lifelike the Mona Lisa looks in this painting. And she looks lifelike because her expression changes as you look at the painting. And I looked at it and I thought, oh my goodness, her expression does change. But because I'm a visual physiologist, I noticed her expression changed depending on where I was looking. So I want you to look at her eyes and think about how much she's smiling. Now shift your gaze so you're looking directly at her mouth. And she seems to be smiling less. Look back and forth between her eyes and her mouth and see if her expression doesn't change systematically depending on whether you're looking at her eyes or her mouth. So what Gombrich says is that Leonardo used sfumato. He blurred it. And that means her expression is ambiguous. But that would mean that her expression ought to vary depending on your state of mind, not where you're looking. Where you're looking is very low level. And as a visual physiologist, I'm very aware that my acuity and your acuity falls off dramatically from your center of gaze. So if you're looking at that dot, all these letters are equally readable. That's how much your acuity falls off with eccentricity. So your central vision is really good at seeing fine details, high spatial frequencies. Your peripheral vision can't see high spatial frequencies. It can't see details. That's why you have to move your eyes when you read, because only your central vision can resolve those tiny letters. But your peripheral vision isn't a bad version of your central vision. It's actually better at seeing big, blurry things. So if you filter the Mona Lisa in such a way that you can see what she would look like to your peripheral vision, she's quite cheerful. But if you filter her in such a way that you can see what she would look like to your central vision, if you could see her whole face with your central vision all at once, which of course you can't, she's not particularly cheerful. So you move your eyes around constantly. You have to. If you don't, your vision fades because your visual system cares about spatial contrast, but it also cares about temporal contrast. So you move your gaze around 2 or 3 times a second. That means that as you move your eyes around the Mona Lisa, her expression changes. And it gives this painting a dynamic quality. And 500 years ago, a dynamic quality in a static painting was something very special. It also gives her a coy quality that Gombrich mentions. So you're looking at the background and she seems to be grinning from ear to ear, and so you try to catch her smiling by looking at her mouth and she stops. I think that the pointillists, the post-impressionists, achieved a vibrancy in their colors for the same reason. Now the pointillists themselves thought that their vibrant colors came about by additive color mixing. So if you mix 2 pigments on a palette, you're mixing their absorbances. Whereas if you put separate colors in little dots, you're mixing their reflectances. So that's additive versus subtractive color mixing. And the pointillists themselves thought that they were achieving additive color mixing, and that's why their colors were so sparkly. But magazine illustrations are made up of little dots of color, and there's nothing special about the colors that you get in magazine illustrations. So I think instead that the sparkly quality of pointillist paintings comes about because your central vision sees dots, your peripheral vision sees the river, the rower, the trees. And as you move your eyes around the painting, different parts of the painting are seen as dots or solid areas. And so that much change, that much difference in what it looks like depending on where you're looking, I think gives it a vibrant, sparkly quality. So for example, if you're looking at this reflection, if you look right at it, you see dots. If you look at this rock, you see stripes. And if you look way over here, it's a solid. And that's what real water looks like, right? Real water sometimes is dots, sometimes is stripes, sometimes is solid, and it sparkles. Chuck Close takes this even further by making large tiles of color. With your central vision, you see the tiles. With your peripheral vision, you see Chuck Close. Photomosaics. With your central vision, you see baseball cards. With your peripheral vision, you see Babe Ruth. So now I want to talk a little bit about higher level visual processing. How do we recognize things? We primates are very good at recognizing faces. In a tenth of a second, you're not only detecting faces, you're recognizing particular individuals. And you're really good at this, and you can do it whether they're cartoons or whether they're degraded or whether they're occluded, and you can do this better than any computer program. Again, in trying to figure out how we do this, artists were way ahead of us neuroscientists, because artists figured out, like thousands of years ago, that a caricature is easier to recognize and seems more like that person than a veridical line drawing. So this looks more like my president than this actual line drawing that was copied from a portrait. Why are caricatures so evocative of a particular individual? Well, we've been studying this using monkeys. Monkeys, like people, use faces to recognize conspecifics and to convey important social information. Humans have special regions in their temporal lobe dedicated to processing faces. We know this because you can get a lesion in your temporal lobe and lose the ability to recognize faces, even though your other object recognition abilities will be intact. Using functional MRI, we can see these regions in people that are specialized for processing faces because they light up when you show individuals' faces versus other objects. Monkeys also have face patches, just like we do. And in my lab, we have recorded from face patches in monkeys. And if you stick an electrode in an MRI-identified face patch, you will find cells that fire in response to faces. And they'll fire in response to people faces, they'll fire to monkey faces, they'll even fire to cartoon faces. Anything you would call a face, a monkey face cell will respond to. So we wanted to know, how do face cells recognize faces? And to do this, we used cartoon faces, and we made a cartoon face set in which we took 20 different variables and varied them as far as we could, having everything we could think of to do with faces, the shape of the faces, the shape of the eyes, the eyebrows, the mouth, the nose, everything. We varied everything we could think of, and we varied it simultaneously so that we could efficiently go through all these faces. And here's the stimulus set we used. We can see that it goes to some pretty extreme faces. And we asked, what do face cells care about? So if each face cell codes a single individual face, we would never have gotten anything. But because we use these parameters, we could say, well, do face cells, in a sense, code these particular variables? And we found that they did. Here's the only data we're going to have here. If you like data, pay attention. These are 3 face cells. Each row is 1 face cell. Each column is the tuning of that face cell to that parameter. So here we've got 2 face cells that care about the size of the iris and 1 that doesn't. And then 1 that cares about the size of the eye but doesn't care about. So you can see that each face cell cares about a few of these parameters that we tried. And they're different parameters for different face cells. So you could get all of the parameters for an individual face by a constellation of face cells with these overlapping parameters. Now, you notice we've got 2 inner eye distance. Where is that? So 2 of the cells cared about inter-eye distance, how far apart the eyes were. And you could imagine that if you wanted to code faces by how far apart the eyes were, which is an important face parameter, you might want to have cells that coded for all possible inner-eye distances. Like 1 cell would be for medium inner-eye distances, 1 would be for a little bit close, 1 would be for a little bit far, and so on. So that's a lot of cells that you'd need. Or you could have 1 cell that liked really big inner-eye distances and then was inhibited by small. And you could have another cell that's excited by little inner-eye distances and inhibited by big. This is more efficient because you get all the in-between distances for free from the ratio of these two. And indeed, that's what we saw. You can see that when a cell is significantly tuned to one of these variables, it tends to be ramp-shaped, that is, peaking at one extreme and inhibited at the other extreme. You can see here again for iris size, inner eye distance, shape of the eyes, and face shape. In general, ramp-shaped tuning was what we saw for all of these variables. Here are a whole bunch, these are just hundreds of cells. Each row is an individual cell, and showing the tuning for each of these parameters. And you can see that for many of them, they peaked at one extreme or the other, peaking being yellow and suppression being blue. And here's the entire population of a whole bunch of parameters, hundreds. And you can see that in general, they peak at extremes. So this ramp-shaped tuning is something we saw again and again in face cells for just about all the parameters we looked at. So that means that what face cells are doing is telling you how a face differs from the average. That is, it only fires when something is different from an intermediate value. And of course, that's consistent with caricature, right? What's a caricature? You take a face, you say, how does it differ from the average, and then you exaggerate the difference. So essentially, face cells are saying, what's distinct about this face, and kind of making a caricature of it. You can see this in the work of artists. These are some portraits by Picasso. Like most Picasso portraits, they look peculiar, but I've put them next to a photograph of the person they're a portrait of. This is Picasso himself, Gertrude Stein, Igor Stravinsky, wife of a friend, girlfriend, art dealer, and mother-in-law. You can see that he has extracted what's unique about each of these individuals. This also means face processing must involve within category comparison, because you can't say somebody's eyes are far apart unless you know what you're comparing it to an average inner eye distance, right? So that means that face processing, like everything I told you about at the beginning of the talk, is local and opponent, like luminance, like color. What do I mean by that? So I told you that as you march across early visual cortex, you march across visual space. But as you march across higher visual cortex in the temporal lobe, you march across categories. So you can record from face selective cells here, and then you move over a little bit and you'll find you're recording from body selective cells. Or you move over a little further and you're recording from place selective cells. So they'll respond to places or bodies or faces all over the visual field. So that means that locally in cortex, that is where inhibition is going to be strongest, is within category. So that means that face processing is opponent, the opponency is within category. So in the same way that you can get an after image from color or an after image from light, you can get an after image in face identity. So what I want you to do while I'm nattering on here is look at these little numbers. The reason I want you to look at the numbers is because I don't want you to get a dumb little retinal after effect. I want you to get a high-level face identity after effect. So you've adapted to these two identities. And now what I'm going to do is show you two identical morphs. And I hope you get a face identity after effect. So face processing is local and opponent. So vision is information processing, not image transmission. You can see this in the work of many artists. You can see it in the art of children. If you ask a 3-year-old child to draw a person, you will get a face on legs, right? The child is telling you that he has extracted the information about how to identify an individual, and most of that information is in the face, right? This is a drawing a child made of her pregnant mother. A face on legs inside a face on legs. Matisse said this explicitly. He doesn't paint things, he paints the differences between things. So here's Matisse's large pink nude, and in a sense, he has extracted the difference between a female body and other objects, or a female body and a male body. What are the distinctive things? And then he's exaggerated these differences. But you know how I mentioned that artists do experiments? They work at this. This is several months, from May through October, that Matisse worked on this drawing. And you can see how he figures out when he's captured one of these abstractions that he wants to incorporate into this painting. And it gets closer and closer to what it is he wants to do. And he doesn't know how he's going to capture this abstraction. He has to try all these different things until he gets it. And when he gets it, he knows it. So I get invited to some strange meetings sometimes. And one of the stranger meetings I got invited to was an exhibition of dioramas at the Museum of Art and Design. You know what a diorama is? Your kids do it in middle school, you know, with the shoebox and the little bits of leaves and stuff, right? But I went anyway, even though I thought it was a dumb idea, and I was enchanted. These dioramas were exquisite. Scenes that would be boring or kind of unpleasant or uninteresting or even downright ugly were exquisite. Now, why were they so interesting? I spend a lot of time trying to talk to artists about what it is they're doing. And we have a language problem. And I asked these artists what they're doing, and they said, I'm trying to get the viewer to see this differently. Now, as a scientist, that didn't mean that much to me when I first heard it, but I thought about it because that's what they kept saying. And I realized they really were getting me to see it differently. And I think that the way they got me to see it differently has to do with the fact that inferotemporal cortex is organized into these domains for special object categories. So we know about the different domains from the kinds of agnosias that neurological patients can get. Patients can lose the ability to recognize faces in particular, or places, or text, or bodies, or tools. So that means you must have specialized regions in your inferotemporal cortex for processing, for recognizing these particular object categories. And of course, with fMRI, we can now see that we do have domains for faces, places, text, bodies, and tools. But why do we have these domains? I think they make us expert at processing these different object categories because they allow us to do fine within category comparisons. Now, we are experts at these things. You probably don't think you're an expert at processing faces, but I'm going to show you how expert you are. You know that this is the Margaret Thatcher illusion, and it was invented by Peter Thomson. And you know what I've done, right? I've manipulated you even know that it's this one. I'm going to give it away, right? I've manipulated the features in this face. But right now, you are processing these faces with your not face cortex, with your not expert cortex. You're using ordinary inferotemporal cortex, random object. You're not using your expert face cortex. And now when I turn this right side up, even though you knew what I did, you see this differently, right? Now you're an expert. That's the difference between your expert processing and your non-expert processing. And I think that when artists make things the wrong size, they do the same thing. They get us to use a different kind of cortex to process it. They literally get us to process it differently. They literally get us to see it differently, using a different part of our brain. Even just putting something in the wrong place. Okay, now, you know what I did. This is exactly the same image. Can you see it the same way you did before? No. Isn't that amazing? Cracks me up every time. Okay, the last thing I want to talk about is stereopsis, which is carried by our wear system. Now this time I want you to use the glasses. Blue on the right, that's stereopsis. Some of you have stereopsis. 10% of otherwise normal people have no stereopsis, but stereopsis contributes to your sense of distance and depth. Stereopsis is just geometry. You have two eyes, they're separated by a few inches, They see the world from slightly different points of view. So if you're looking at an object, it casts images on corresponding parts of your two retinas, but objects that are closer or farther away cast images on non-corresponding parts of your two retinas. So if you go to a museum and you find a painting where the artist has obviously gone to a lot of trouble to include cues about distance and depth, like shading and perspective and so on, those cues give you a sense of depth. But your stereo, if you're looking at it with two eyes open, is saying, no, sorry, it's flat. And so you're working against the cues that the artist has given you. So if you want to fall into a painting, next time you go to a museum, get a big one and close one eye. And it will seem much more three-dimensional because your stereo is no longer telling you that it's flat. But stereo matching is a computationally difficult problem. So if I tell, no glasses, no cheating, come on. Let's see if you can figure this out. The blue checks are white to your right eye, the red checks are white to your left eye. You're a smart audience. Can you figure out what it shows? No, probably not, but your visual system can solve it instantly. Blue, both eyes open, blue on the right, and you will, some of you, see something. Some of you won't see anything, don't worry, okay? Really, it's not important. If you have 4 identical objects out there in the world, they cast images on corresponding parts of your two retinas, and you can reconstruct the correct answer as being this, but you can misfuse number 3 in the right eye with number 4 in the left eye, and you will get the same object just at a different distance. So have you ever misstepped on an escalator, which have those slats, you know? You do that because if you misfuse these identical slats in the escalator, it corresponds to the same step, just at the wrong position. And I think some artists achieve a sense of depth. I think the Impressionists who were painting the air did so by having dabs of paint that you could misfuse in your two eyes that would correspond to a volume of speckles. And I don't even like looking at this one because it has that kind of a three-dimensional quality that I find a little unpleasant. Because you can misfuse the leaves in one eye with the same leaf in the other eye. But what's interesting about this is the guy who painted it, Gustav Klimt, didn't have stereopsis. How do I know he didn't have stereopsis? How do I know? Oh, come on. He's wildly cross-eyed. He couldn't possibly have stereopsis. So stereo is computed in primary visual cortex, where the inputs from the two eyes first come together. And primary visual cortex has a very precise map of visual space. And if your eyes aren't lined up, the same part of visual cortex isn't dealing with the input from the same part of the visual field in the two eyes. So you can't possibly compute stereopsis. I got interested in the idea that artistic talent and reading difficulties might be correlated. So I hope you find this unpleasant to read. Do you know what dyslexia is? It's a problem learning to read. So there are people of normal intelligence with normal educational experience who have a terrible time learning to read. And they sometimes complain that black and white text has this same jittery, slidey, unpleasant quality that I see with equiluminant text. So we did some studies on dyslexics, and we found that there was we measured the evoked potential. It's kind of an EEG to low contrast checkerboards. And we found that the dyslexic subjects had a slightly slower peak to this low contrast stimulus than control subjects. So a slight slowing in the dorsal stream pathway. Now remember, what does the dorsal stream do? Motion, depth, spatial organization, and figure ground segregation. It doesn't say reading, it just says these kind of functions. But what are the other functions for the dorsal? What are the other symptoms of dyslexia? Positional information problems, figure ground problems, and trouble gauging distance and depth. So in the process of doing these studies, I was privileged to meet a number of people with dyslexia. And a surprising number of them were artists, musicians, computer programmers, and actors. And it's been documented that dyslexics are overrepresented in the artistic population, and the usual explanation is they were no good at academic subjects, so they spent all their time in the art room. But I was so impressed by how talented some of these people were, I started thinking, well, maybe there's something about the dyslexic brain that makes them better at art and music than the rest of us. So a slight slowing might explain some of the auditory differences that you see. So the difference between ba and da is a very rapid auditory transition. It takes 40 milliseconds. So that might explain why dyslexics have trouble with phonetics. They can't hear the difference as well as control people. But music is all about slow auditory transitions. Maybe if they're worse at fast, they're better at slow. But I don't work on the auditory system, I work on vision. So why are there so many dyslexic artists? How about the idea that trouble gauging distance and depth is an asset to an artist? So I can't draw a chair. If I look at a chair, my visual system extracts all this three-dimensional information about it, and I can't flatten it again in order to draw it. So maybe a kid that sees the world as flatter than his companions is going to be better at drawing whatever it is he sees on a flat piece of paper. So this is Chuck Close. His eyeglasses are different strengths in his two eyes. He's anisometropic, he's severely dyslexic, and he says he sees the world as flat. There's a clinical ophthalmological test for whether you're, did I mention this? 10% of otherwise normal people have no stereopsis. There is a clinical test for telling whether somebody's eyes are lined up, which is to look at the light reflection in their two eyes. And if the light reflection is in exactly the same place in their two eyes, they could have normal stereopsis. So you see the light reflex in the same place in the two eyes? See it? Are you good at this now? Okay. There is one baseball player who had no stereopsis. Light reflexes center in that eye, off to the side in that eye. Babe Ruth had amblyopia. As Gunnar mentioned, David Hubel and Torsten Wiesel figured out that if you close one eye or you have the two eyes misaligned, the brain gets rid of the inputs from one of those eyes, and essentially, if this happens when you're young, you become blind in one eye. That's called amblyopia. That's what Babe Ruth had. So we looked at a bunch of artists. Oh, I forgot to say this. I told you 10% of otherwise normal people have no stereopsis, but only 3% of the population has significantly misaligned eyes. It's not that common. So you can have no stereopsis for some unknown reason, or you can have no stereopsis because your eyes aren't lined up right. So you'd expect out of maybe 100 famous artists that we looked at, we would have found one or two. We found 16 right off the bat. This is Andrew Wyeth. This eye is looking at you. This eye is looking out that door. His father, N.C. Wyeth, was an illustrator. His eyes also deviate outwards, and I'm hoping someone is going to send me a good photograph of Jamie Wyeth. Edward Hopper, the light's next to his pupil on this side and off at the edge of his iris on that side. Kara Walker, every picture I've ever seen of her, the light reflexes in two different places in her two eyes. Kara Walker works in silhouette. How flat can you get? I don't know about the dog. Marc Chagall was cross-eyed. Frank Stella was cross-eyed. Jasper Johns wall-eyed. His eyes deviated outwards. Robert Rauschenberg was wall-eyed and severely dyslexic. Alexander Calder was wall-eyed. Man Ray. Frank Lloyd Wright. Now, I told you it's a legitimate clinical test to take a picture of somebody and see whether their eyes are misaligned. You're not really allowed to do this with a painting for obvious reasons. But I was at the Louvre once, and there's a room, a little tiny room. All it has in it is 4 Rembrandt self-portraits, one on each wall. And all 4 of them, one eye is looking straight ahead and the other eye is deviating off to the side. Now, I know that when you paint a self-portrait, you look in a mirror. And when you're painting your right eye, you look to the right. When you're painting your left eye, you look to the left. So this is a montage of me. And on this side, I'm holding a mirror about 8 inches from my nose. On this side, I'm looking at my right eye. On this side, I'm looking at my left eye. And my light reflexes in slightly different places, even though I do have stereopsis. But my eyes aren't deviating nearly as much as Rembrandt portrays himself. So I had this pile of Rembrandt self-portraits, reproductions of Rembrandt self-portraits on my desk, and my young colleague came into my office and said, "Is it always the same eye?" And I said, "No, if it were, I could publish it. Sometimes it's the eye on this side that deviates, and sometimes it's the eye on this side." My young colleague was himself a stereo-blind artist, and he said, "Marge, you have to separate the paintings and the etchings." And when we did, we discovered that in the paintings, it's always the eye on this side that deviates. In the etchings, it's always the eye on this side that deviates. The way you make an etching is to scratch something on a plate and flip it over. So it wasn't a trope. Rembrandt was not trying to achieve a particular image where he's looking off into the something or another. Instead, he was probably drawing exactly what he saw when he looked in a mirror. So we took a Photoshop and we outlined where the eye was and where the iris was and all the eyes, and we made a graph. So here's a take-home message for a scientist. If you can make a graph of something really unlikely, you can get it published. This is the only thing I have ever had published in the New England Journal of Medicine. In general, in the paintings, the eye on the right looked straight ahead. The eye on the left deviated outward. This is the one that's in Berlin. We think it's either a fake or he painted it from an etching. And in the etchings, it's the reverse. So this doesn't mean that if you want your kid to be a great artist, you should poke out one eye. I just think it lends a kid an advantage that he's probably not aware of why he's got this advantage. But the first thing you learn in drawing class, close one eye. So, don't poke out anybody's eye. And here, if you want some take-home messages, the really important one is vision as information processing. You have different subdivisions in your visual system, and they do very different things. Our evolutionarily oldest visual abilities are colorblind. You don't have to color inside the lines, and people are different. And some people may be good at one thing, and their brains may be different and makes them good at things that you're not so good at. And if you would leave the stereo glasses somewhere near the back on your way out, I would be grateful, and I'm happy to take questions.